Good morning, everyone. The topic of my presentation is biology of memory, and my moderator is Dr. Shobit Garg, sir. He will be studying about the introduction, types of memory, neural plasticity, long-term potentiation, cortical organization of memory, and assessment of memory. Introduction. Memory is the glue that binds mental life, the scaffolding for personal history. Personality is in part an accumulation of habits that have been acquired many very early in life, which create dispositions and influence behavior. In the same sense, the neuroses are often products of learning, anxieties, phobias, and maladaptive behaviors that result from particular experiences. Psychotherapy itself is a process by which new habits and skills are acquired through the accumulation of new experiences. In this sense, memory is at the theoretic heart of psychiatry's concern with personality, the consequences of early experience, and the possibility of growth and change. Memory is the ability to store, retain, and retrieve information. Our memory stores our personal experiences, emotions, preferences or dislikes, motor skills, world knowledge, language. Fundamentally, we as a person are derived from experiences that have been stored in our nervous system. Processing of memory consists of encoding, that is coding and putting into memory, storage, that is maintaining the memory, and retrieval, that is recovery from memory. These are the types of memory which have already been discussed. The stimuli comes which inputs and it forms the sensory memory which can be forgotten, and if attention is paid it gets converted into short-term memory. Then the encoding occurs and it gets converted into long-term memory, and on retrieval it again gets converted into short-term memory. Now, the structures that are involved in memory. Hippocampal formation that consists of the dentate gyrus, the hippocampus, and the subicular complex and linked regions of medial temporal lobe with prefrontal cortex play a critical role in encoding and retrieval of episodic memory. Diencephalic structures like medial thalamus, mammillary body, and fornix also play a part in memory. Fair conditioning and extension, it occurs in interaction between amygdala and cingulate gyrus. Basal ganglia and cerebellum is important for procedural memory, and neocortex is the ultimate store of the memory. Synapses to memory. Memory is a special case of the general biological phenomena of neural plasticity. Neurons can show history-dependent activity by responding differently as a function of prior input. The plasticity of nerve cells and synapses is the basis of memory. Experiences can lead to structural changes at the synapse, including alterations in the strength of existing synapses and alterations in the number of synaptic contacts along specific pathways. This is the neural plasticity, which has been discussed, and long-term potentiation, which has also been discussed. The cortical organization of memory. The functional organization of the mammalian cerebral cortex has been revealed through neuropsychological analysis of deficits following brain damage and through physiological studies of intact brains. The cortical area is responsible for processing and storing visual information has been studied most extensively in non-human primates. The cortical pathways for visual information processing begin in primary visual cortex and proceed from there along parallel pathways or streams. One stream projects ventrally to inferotemporal cortex and is specialized for processing information concerning the identification of visual objects. Another stream projects dorsally to the parietal cortex and is specialized for processing information about the spatial location. Specific visual processing areas in the dorsal and ventral streams, together with areas in prefrontal cortex, register the immediate experience of perceptual processing. The results of perceptual processing are first available as immediate memory. Immediate memory refers to the amount of information that can be held in mind, like a telephone number, so that it is available for immediate use. Immediate memory can be extended in time by rehearsing or otherwise manipulating the information, in which case what is stored is now said to be in working memory. In sum, memory is distributed and localized in the cerebral cortex. Memory is distributed in the sense that, as Lashley concluded, there is no cortical center dedicated solely to the storage of memories. Now, memory and acetylcholine, two sets of acetylcholine projections, one arising from the brainstem neurotransmitter center and the basal forebrain, basal nucleus or nucleus basalis of Maynard, as well as the medial septal nucleus. These cholinergic fibers play a prominent role in memory. Nucleus of Maynard have the principal role in mediating memory function. Short-term memory disturbance of Alzheimer's patient is due to degeneration of these cholinergic neurons. In Alzheimer's patient, short-term memory disturbance is due to degeneration of the cholinergic neurons. Other cholinergic neurons, such as in the striatum and projecting from lateral tegmental area, are not involved in the memory disorder of Alzheimer's disease. Cholinergic deficiency, part of vascular dementia or alcoholic dementia. This is why some patients with vascular dementia or alcohol-related dementias respond to cholinesterase inhibitors. The amnesia part has also been discussed. Sleep and memory. Freud noted that dreams can reveal fragments of recent experiences in the form of day residues. Although many questions about how and why memories may be processed, Although many questions about how and why memories may be processed during sleep remain unresolved, recent experiments have provided new empirical support to bolster the idea that memory processing during the sleep serves as adaptive function. Memory storage appears to be specifically aided by processing during deep sleep within a few hours after learning, especially in stage 3 and 4, slow wave sleep. Some results indicate that slow wave sleep facilitates the storage of declarative memories, but not non-declarative memories. Now, frontal lobe and memory. Amnesia does not occur after limited frontal damage. The frontal lobes are fundamentally important for declarative memory. Patients with frontal lesions have poor memory for the context in which information was acquired, difficulty in free recall, and even have some mild difficulty on tests of item recognition. Patient BG suffered an infarction restricted to the right frontal lobe, resulting in substantial false remembering. He had an abnormal tendency to claim that some stimuli were familiar, even though not being presented for study. His false responses probably relied on a general feeling of familiarity for the kind of stimuli that has been presented, rather than on specific memories for the stimuli. Psychogenic amnesia, also known as dissociative amnesia or functional amnesia. It is characterized by abnormal memory functioning in the absence of structural brain damage or a known neurobiological cause. It results from the effects of severe stress or psychological trauma on the brain. New learning capacity is not affected. Patients with neurological amnesia never forget their names, their remote memories for the events of childhood and adolescence are typically normal. Some patients with psychogenic amnesia have circumscribed retrograde memory loss that covers a particular time period or that covers only autobiographical memories. ECT procedures produces a transient amnesia manifested by a diminished ability to form new memories during the period of treatment. The amnesia remits within days or at most a few weeks after completion of treatment. The patient is left with a retrograde amnesia for many events during the days or weeks of treatment. Korsakoff syndrome has been discussed. Now, the assessment of memory. A complete assessment of memory usually involves assessment of intellectual functions, new learning capacity, remote memory, and memory self. New learning capacity. Two important principles of new learning capacity are: tests are sensitive to memory impairment. Information is presented that can be held in immediate memory. Example, paired associate task. Tests are sensitive to memory impairment when a delay filled with distractions interposed between the learning phase and the test phase. Memory can be tested by unaided recall of previously studied materials, free recall, by presenting a cue for the material to be remembered, cued recall, or by testing recognition memory, yes, no recognition tests, multiple choice tests. Remote memory can be tested by autobiographical memory tests, word probe tasks. Patients are asked to recollect specific episodes from their past in response to single word cues, for example, bird and ticket, and to date the episodes. Test about material in the public domain, example about former one-season television programs, news events, or photographs of famous persons. Normal subjects, most of the memories of recent time periods, the past 1 or 2 months. Patients with amnesia, few episodic memories from the recent past, but many remote autobiographical memories as normal subjects. Now, the memory self-report. Patients can often supply descriptions of their memory problems. Tests used are called tests of meta-memory. Depressed patients rate their memory as poor in a rather undifferentiated way, endorsing equally all the items on a self-rating form. Amnesic patients endorse some items more than others. There is a pattern to their memory complaints. Amnesic patients do not report difficulty in remembering very remote events or in following what is being said to them, but do report difficulty remembering an event a few minutes after it happens. So, the next. So next group discussion is a mental status exam.
Anyone who wants to ask question? Anything? Dr. Arpita will give extra marks. Anything with EMPA receptors? EMPA, so how this long-term depression potentiation takes place? What is the difference? Why a neuron decides to potentiate होगा, a neuron decides to depression. कैल्शियम जो फ्लो है वो फास्ट है तो पोटेंशियल डिप्रेशन होगा, स्लो है तो डिप्रेशन। एएमपीए रिसेप्टर और बनेंगे, है ना, तो जाके सिनेप्स में लगेंगे, डिप्रेशन होगा। तो उसमें your post-synaptic neurons will be activated after your pre-synaptic. Reverse will happen. Post-synaptic neuron is first activated, then pre-synaptic neuron का depression होगा। इसमें फिर एएमपीए रिसेप्टर्स की एंडोसाइटोसिस हो जाएगी और सिनेप्टिक स्ट्रेंथ कम हो जाएगी। So basically, and there are different kinds of neuroplastics. In fact, in long-term depression, there are 3 or 4 kinds of neuroplasticities and different mechanisms. A whole different topic in itself. 2 to 10 questions, 3 to 2, 3 to 1, 1 to 2. Okay. Declare the minutes. Why we are doing this? Not to say why keep first.
So, you also asked questions and Asta also presented them. You get the charge. मेल कर दो, मेरी से कर दो फिर।